Welcome to the TOEFL listening section. In this part, you will hear one or more conversations in an academic or campus context, such as a discussion between a student and a professor, or between two students. You may take notes while listening. Listen to part of a conversation. You will hear it only once, so listen carefully. After the conversation, you will answer questions about what you heard. Some questions will ask you about main ideas and details. Others will ask about implied meaning, speaker attitude, or how ideas relate to one another. Use both what is stated and what is implied to answer.
Good afternoon, Dr. Martin. Thanks for meeting with me during office hours. I wanted to go over my project on viral evolution tracking and get your feedback on the sequencing strategy.
Good afternoon, Javier. I read your proposal briefly. So tell me what you plan to sequence and what you hope to identify about viral evolution.
I want to focus on genomic sequencing for variant identification in respiratory viruses, specifically tracking emergence of amino acid changes in the spike protein and mapping their spread across our region.
That is a clear focus. Are you planning amplicon sequencing or a metagenomic approach, and why?
I was thinking amplicon sequencing with tiled primers because it gives high read depth across the genome, which improves variant calling, but I worry about primer dropouts with divergent variants.
Exactly. Amplicon sequencing gives excellent read depth and is cost-effective for large surveillance, but you must monitor for primer mismatches. Metagenomic sequencing avoids targeted primers and can detect unexpected recombinant segments, but it is more expensive and often gives lower coverage.
When you say recombinant segments, do you mean recombination between strains that could confound lineage assignment?
Yes, recombination can create mosaic genomes, so a simple tree may place different segments in different positions. We screen alignments for recombination signals before inferring phylogenies to avoid misleading topologies.
That makes sense. For variant calling, I plan to set a minimum read depth threshold and a variant allele frequency cutoff to call single nucleotide polymorphisms and indels. Is that standard?
Yes, it is standard to require sufficient read depth and an allele frequency threshold, often around 0.7, for consensus calling in surveillance. For intra-host diversity studies, you might lower that threshold carefully, but then you need strict error models.
I also wanted to ask about lineage assignment. I know tools like Pangolin assign lineages, but how reliable are they when new mutations appear rapidly?
Pangolin is useful for assigning known lineages, but it depends on the reference dataset. Rapid emergence of mutations can produce novel lineages that need curation. Visualizing sequences on Nextstrain helps see placement and detect clusters that lack formal lineage names.
Could you give an example where this happened in real surveillance work?
Take the early SARS-CoV-2 waves. Delta and later Omicron accumulated many spike mutations quickly. Some mutations arose independently in different lineages, an example of convergent evolution. That made automated assignment tricky until experts designated new lineages.
You mentioned convergent evolution. Does that mean similar selective pressures led to the same spike changes in unrelated lineages?
Exactly. Immune pressure or transmission fitness can select for similar substitutions in the receptor binding domain across different viral backgrounds, leading to convergent changes.
How do we use phylogenetic trees to track transmission chains and to date when a variant emerged?
We use time-stamped sequences and apply a molecular clock model to estimate substitution rates. With phylogenetic methods, we can infer when the most recent common ancestor of a cluster existed, which provides an estimate of the date of emergence.
Are there pitfalls in dating estimates that I should be aware of?
Yes, sampling bias can skew estimates. If you only sequence samples from a few locations or from a particular time window, the inferred origin or growth rate may be incorrect. Low sequence diversity or sequencing errors also reduce the precision of molecular clock estimates.
What about integrating non-clinical sources like wastewater surveillance in tracking viral evolution?
Wastewater sequencing is a powerful supplement. It captures pooled viral genetic material from a population and can reveal rise of variants prior to clinical detection. However, it often yields mixed signals and lower genome completeness, so you need careful deconvolution methods.
For public health impact, what calculations do we present to authorities when we identify a new variant?
We report genomic features such as spike mutations of concern, lineage assignment, estimated date of emergence, and phylogenetic metrics like effective reproduction number changes if supported. We also include uncertainty intervals and caveats about sampling limitations.
On data sharing, which repository should we upload sequences to for maximum utility?
GISAID is the primary platform used for rapid sharing in respiratory virus surveillance because it preserves metadata and facilitates global analyses. We also deposit raw reads in public archives when permitted by consent.
Thanks, that helps. For my methods section, I will explain the sequencing platform, amplicon design, read depth thresholds, lineage assignment with pangolin and tree building, with time-stamped sequences and recombination screening.
That sounds thorough. Also include a paragraph on quality control steps, such as trimming low-quality bases, monitoring primer mismatches, and validating unusual mutations with replicate sequencing.
I will. One last question about convergent mutations. How do we distinguish true convergent evolution from contamination or artifacts?
We check raw read alignments to ensure variant support across reads and replicate samples. We also look for consistent phylogenetic signal and rule out lab contamination by tracking sequencing batches and negative controls.
Great, I will draft the revised proposal and include those controls and the plan to upload to GISAID. Thank you for your guidance.
You are welcome, Javier. Send the draft, and we will refine the analysis pipeline and the phylogenetic models together.
Listen again to part of the conversation, then answer the question.
We use time-stamped sequences and apply a molecular clock model to estimate substitution rates, which helps us date when a variant emerged.
This concludes the conversation portion. For more TOEFL practice tests and study materials, visit Spinta.org.